Okay, next we will continue with the autosomal linkage and looking at how it can be applied to find the crossover frequencies between two genes and also the distance between the two genes in the chromosome map. Alright, so when you look at this pair of homologous chromosomes, right, there are three genes, right, in this pair of homologous chromosomes. We have the gene A, gene D, and gene B. The distance between gene A and D is larger, right? The distance between D and B. Alright, so what you can see is when the distance between the two genes on the same chromosome is larger, right, or they're further apart, there is higher chance of forming chiasma between the two genes. Right, so again, crossing over is random, right? So like anywhere along the chromosome, you can expect to form chiasma or chiasmata. Okay, so the more distance there is between the two genes, right, then more likely that the chiasma will be formed between the two genes. So when we talk about crossing over, right, from the first video we have been emphasizing this, crossing over may or may not occur, number one, and if it occurs, right, it may not cause the crossing over between the two genes, right? So in order for you to have crossing over between two genes, the chiasma must form between the two genes. Okay, so having a longer distance between the two genes means there's a higher chance of forming that chiasma between the two genes, and therefore higher chance of crossing over between the two genes. Alright, as we have shown here. Okay? So, in order to form the recombinant gametes that have a new combination of alleles between these two genes, right, a longer distance between the two genes would increase the chance of forming that recombinant. Alright, so in this case, our chiasma is formed between gene A and D. Because they're longer, right? There's further distance, there's more chance for the chiasma to be formed. Unlike between gene D and B, right? Which is such a short distance, right? Then it is a lot as likely, right, for the chiasma to be formed between these two genes. All right. Okay, so I want to introduce you to a term called tightly linked or complete linkage. That's used to, these two terms are used to describe genes which are very close together, or sometimes in the wording of the question, they might say they are next to each other. Right, so when they are so close together, like I say, right, then the chance of having the chiasma between the two genes is low, and therefore the chance of forming recombinants is low. Right, so when they are so close together, crossing over is not likely to occur between these two genes. Right? So the alleles on the same chromosomes for two tightly linked genes would always be inherited together. So in this case, if I take gene D and gene B to be tightly linked, right, then your small d and big B would always be inherited together, and your big D and small b would always be inherited together. There's no chance for big D, big B, or small d, small b, because there's no chance for crossing over if I consider these two to be tightly linked. Understand? Okay? So that's a term that sometimes you see in questions when they say tightly or completely linked. Then when you see that, you know, right, there shouldn't be crossing over. Alright? Okay, so the chance of crossing over, or the proportion of crossing over, depends on the distance between the two genes, right? So therefore, when we look at the proportion of recombinants that are formed, which comes from crossing over, we can deduce the distance between the two genes. Okay, so the proportion of recombinants formed, that's called the crossover value, or we use it to calculate the crossover value, which means basically is a percentage of recombinants that are formed. Okay, so 1% of this percentage would equate to 1 centimorgan on the chromosome. Centimorgan is the unit for distance between genes on a chromosome. Alright, so 1% equals to 1 centimorgan. Okay, so I have an example in the notes on how to calculate the COV and how to deduce the distance between the genes, right? So can you just read the example yourself, so I don't have to like just read off from the notes? I know some of you don't like the lecturer just reading from the notes, right? So can you just look at the example yourself?